Saturday looms, right? And we've only got a 2-day weekend this weekend. I mean, what's the thinking with that? Not good, right? Turn this volume down just a little bit and see if that helps. There we go. Okay. A lot of energy in here today. Who said that? All right, dumb jokes today, right? All right, so last time I went through some basic structural considerations for lipids. Today I'm going to talk a little bit more about those structural considerations for some other lipids, and also how those structural considerations play into the properties of membranes. Because the reason we're talking about these is we need to begin to understand something about the structure of membranes. Because membranes, as we will see, the integrity of membranes is absolutely essential not only for a cell, but for all the organelles of a cell as well. And so having good membranes that are appropriate for the environment in which they're found is really essential for a cell, because without that, the cell is dead in the water. Well, last time I started talking about this group of molecules, and I went very quickly and briefly through it, so I want to say a little bit more about them today. These are the group of molecules that I want to talk about here. The big category were the icosanoids. And icosanoids, you'll recall, are molecules that have 20 carbons. They are derived from arachidonic acid. And this figure is actually a little confusing in that respect. So I want to make sure that we're clear on what this figure is actually telling us. Okay? So the primary group of molecules we talk about in this category are a group of molecules we call the prostaglandins or the prostanoids. And there's actually a slight difference in what each one of those means. Prostaglandins is a specific group of molecules. You see two examples here on the left, prostaglandin H2, prostaglandin F2 alpha, and there are many other prostaglandins. These are molecules that are made from arachidonic acid. They do have 20 carbons, and they have some interesting physiological properties in the body. Some of them are linked to pain, for example. So prostaglandins, I actually have a song about prostaglandins, which we're not going to sing today. We'll probably sing it next time. But a song about prostaglandins that talks about that role in pain. And so one of the strategies for avoiding pain is to avoid the production of prostaglandins. And how do we avoid the production of prostaglandins? Well, the enzyme that makes prostaglandins, I mentioned last time, the enzyme that makes them catalyzes the conversion of arachidonic acid into the various prostaglandins. And I pointed out that this enzyme was called a cyclooxygenase. Okay? It comes by several names. Cyclooxygenase is one name. Another is prostaglandin synthase. Same thing. Okay? And for abbreviation purposes, most people refer to them as COX enzymes, C-O-X. Okay? So COX enzymes are involved in production of prostaglandins. Some, not all, some of the prostaglandins are involved in pain. So if you can avoid the production of prostaglandins, you can avoid some types of pain. And that means that if you inhibit the cyclooxygenase that makes prostaglandins, you have a pain reliever. Well, it turns out that aspirin is a very good inhibitor of the COX enzymes. So aspirin is a pain reliever because of that. Aspirin is something that we refer to as a non-steroidal anti-inflammatory drug. Long name. You're more likely to abbreviate it NSAID, N-S-A-I-D. NSAIDs are, as their name suggests, they're not steroids. They're involved in pain relief, and what they're doing is they're inhibiting the production of prostaglandins. Ibuprofen is also an NSAID. Okay? There's a variety of NSAIDs that are out there, and they fall into two categories. All right? One category, what are called COX-1 enzymes, because they inhibit cyclooxygenase 1, and others are called COX-2 enzymes because they inhibit cyclooxygenase 2. Well, cyclooxygenase 1 tends to be more prevalent. Cyclooxygenase 2 tends to be more localized. And it was originally thought that if you inhibited only the cyclooxygenase 2, you wouldn't have some of the side effects that happen with the cyclooxygenase 1 enzymes. What are some of the side effects? Well, some people don't tolerate aspirin very well because they associate it with stomach problems. Okay? And stomach problems happen because, and this doesn't happen to everybody, by the way. Okay, I take aspirin. I take an aspirin every day. I have no problem with aspirin of any sort. Okay, but some people do have problems with that, and that's because some of the prostaglandins are involved in maintenance of intestinal tissue. So if you're very sensitive to that, you're going to be having more problems with maintaining intestinal tissue, and the most rapidly turning over tissue in your body is intestinal tissue. So if you're inhibiting production of intestinal tissue by inhibiting some of the prostaglandins necessary for that, you're going to have stomach problems. Okay? It took a long time for people to realize that. Some people used to say, oh, well, aspirin, it puts this acid in your body, et cetera, et cetera. Okay? Your stomach is so full of acid that one little aspirin has zero effect on that. Okay? And it turns out that the effect isn't from the acidity. It's actually from the inhibition of prostaglandins. Okay? So when they came out with COX-2 enzymes, they thought, hey, we've got a magic bullet. It's going to stop pain, but it's not going to have some of the side effects that aspirin has. And by the way, ibuprofen is a COX-1 enzyme, okay, like aspirin, right? But they quickly discovered that there were some major problems associated with that because it was actually associated with some heart valve problems. Okay? So a lot of the COX-2 enzymes that drug companies spend billions of dollars investigating, they had to withdraw from the market because of those problems. There's still a couple of them that are on the market because they've been deemed safer and they're used for some very specific circumstances. But that was some of the problems with COX-2 enzymes versus COX-1 enzymes. Okay, well, I brought this figure up and I said I needed to explain a couple things, so let me explain a couple things about this. I've been talking about the prostaglandins. Prostaglandins are involved in a lot of things. Some of them are involved in uterine contraction, okay? Some of them are involved in blood pressure maintenance, okay? Some of them cause blood pressure to go up. Some of them cause blood pressure to go down, right? So they're a confusing group of molecules and they act sort of like hormones. We call them localized hormones because they usually act very close to the place where they're released. And that's the reason we call them a localized hormone. Because hormones in general travel extensively in the body and have effects long ways away from where they're made. Prostaglandins don't do that because they tend to be very unstable and broken down readily in the body. So they usually work in a very localized region. If you get a bee sting, for example, the reason you swell, okay, is partly due to prostaglandins favoring that swelling process and the pain associated with that as well. Okay? Now, let's see, what I was going to say one other thing about that. I'll think about it in a second. The other, you notice another group up here that says thromboxane, okay? Thromboxanes are also prostanoids. And the reason that thromboxanes are prostanoids is because they are synthesized from prostaglandins. So we put them under that big category of prostanoids. Prostanoids include the prostaglandins, and they include the thromboxanes, because they're all derived directly from arachidonic acid. Okay? Thromboxanes, as you can see here, are involved in the blood clotting process. When you take aspirin to thin the blood, it doesn't really thin it, but to thin the blood, when people use that term, they're talking about inhibiting blood clotting. And the way that aspirin inhibits blood clotting is by inhibiting the production of prostaglandins that are necessary for the production of thromboxanes. So if you inhibit the production of thromboxanes, you reduce the likelihood that blood will clot. Okay? So that's one of the reasons that people take aspirin, is for that purpose. I'll actually talk about another reason later in the lecture today, about another molecule. But this is one way of quote-unquote thinning the blood. All right, the last group here, the Leukotrienes, are also involved in blood clotting. They're also involved in things like asthma. Okay? They also have roles in swelling and inflammation. Okay? But they are not in the prostanoids. All right? These guys are not made from arachidonic acid. They're actually made from linolenic acid. All right? And they do have 20 carbons, but they're not in the same, they're not in the prostanoid group. So leukotrienes are in a different category. Make sense? Investigating the synthesis and inhibition of synthesis of leukotrienes is an active area of research for people who are looking for ways of controlling asthma. Okay? And as somebody who's asthmatic, I can relate to that and appreciate that. Okay. Well, I also mentioned very briefly last time the cannabinoids. And cannabinoids are another group of molecules that are icosanoids. They're made from arachidonic acid, or at least some of them are. And they have interesting structures. If you look at anandamide, which is an endocannabinoid, what's an endocannabinoid? When we use that term, an endocannabinoid is something that the body synthesizes to bind to a cannabinoid receptor. We have cannabinoid receptors in our brain. We have cannabinoid receptors in other places in our body. The places that they're most visible are the effects that they have on brain. We didn't know about the endocannabinoids until we began investigating how it was that marijuana worked. The high that you get from marijuana. Okay? You get high in marijuana because this guy, or guys related to it, tetrahydrocannabinol, which comes from marijuana, binds to the same receptor. And it changes the response a little bit, and so you get a feeling of euphoria, giggling, high, munchies, all of these things, because these receptors are associated with, commonly, things like appetite, food intake, okay? And so if you get high and you get munchy, which does happen with marijuana a lot, it's because of that. This group of molecules that are not, they're not made by the body, they're called phytocannabinoids because that means they come from plants. But they're similar in structure enough, and I know that doesn't look much like structure, but they're similar in structure enough that they can bind to the same receptor, okay, and stimulate it in some way. It has a different effect than the endocannabinoids do. People have heard of runner's high? Runner's high happens because that same receptor that's binding this guy is binding this guy, and your body's making this, okay, to help you reduce pain. So one of the reasons that marijuana is actually used to treat people who have issues of pain is because, again, that same receptor, that endocannabinoid receptor, is binding these various compounds. And these aren't just exotic compounds that bind to this receptor. Something virtually everybody in here likes binds to that receptor. And one of the reasons you like it is because it binds to that receptor, and it's chocolate. Yeah, craving for chocolate, right? Why do you crave chocolate? Why do you get that high from chocolate? Now you begin to understand something, right? Okay, kind of cool. Kind of cool. So that's what's happening with that cannabinoid receptor. So endocannabinoids are made by the body. They bind to the endocannabinoid receptor. Phytocannabinoids are made by plants, and they also bind to the endocannabinoid receptor when we ingest them in some way. Okay. Now, oh, by the way, I meant to mention also, this is kind of cool. This synthesis of the, I did the wrong thing here. The synthesis of the endocannabinoid, the anandamide that you see on here, is actually very simple. That's arachidonic acid going from this guy all the way around. And that little piece right there is ethanolamine. So you simply attach an ethanolamine to arachidonic acid, and you've made anandamide. Very, very simple synthesis. Okay, let's see. Let's talk about another class of molecules, and this class of molecules is also important for membranes, at least the prime member of it is, and that's cholesterol. Cholesterol, as we will see, binds in the lipid bilayer of membranes. And the effect that it has on the lipid bilayer of membranes is to widen the transition temperature. And I'm going to talk about the transition temperature later, so I won't talk about that at the moment. But just keep in the back of your mind that cholesterol widens the transition temperature. You can think of it as melting point if you want to. Okay? It doesn't change the melting point, it widens the range over which that melting occurs. So it's widening that transition from a solid phase to a liquid phase. That's what cholesterol does in the membrane. Now, cholesterol is not a trivial component of membranes. If you take brain tissue and you dry brain tissue down, get rid of all the water, the dry mass of brain is 16% cholesterol. This is why your body makes cholesterol. It's very important for membranes. People tend to think of molecules in simple ways. Good, bad, right? That's not how things work. Essential, right? Cholesterol is essential for us. That's why our body makes it. And yes, there are things that are associated with cholesterol that are problematic, like atherosclerosis and other things that we'll talk about later, okay? But it's also important for things like membranes. In addition, cholesterol, as we will see, is a precursor of the steroid hormones, and it's a precursor of the bile acids. Bile acids are essential for digestion. Okay? Well, plants don't make cholesterol, but plants make related compounds, and phytosterol is a related compound. Now, I'm not going to ask you to draw cholesterol, but I do think if I showed you something that looked generally like this, you would look at this and say, that's a cholesterol-related compound. It's very simple. It's got 4 rings. All the cholesterol-related compounds are like that. Okay? And we'll say more about those. Now, one of the things I do want to say something about related to cholesterol are actually where am I at here? The steroid hormones. There we go. The steroid hormones. Okay? So the steroid hormones are made from cholesterol. These include the sex hormones, the androgens, and the estrogens. Androgens being the male associated hormones, estrogens being the female associated hormones, of which estradiol is the most prominent, and of which testosterone is the most prominent androgen. Okay? These molecules are made from cholesterol. It turns out that cholesterol first makes a progesterone-like molecule, and that progesterone-like molecule is made into all of the others. Okay? And if we look at the flow of synthesis, one of the things about the flow of synthesis is that the estrogens are made from the androgens. Now, why do I say that? That turns out to be important because there's a very unusual thing that happens in the synthesis of the estrogens from the androgens. And if you look at it, you can actually see it. You see here a single double bond, and down here you see a benzene ring. This is an aromatic structure. This is a non-aromatic structure up here. Okay? This type of catalysis, this is the only place it occurs in biology where the cell directly catalyzes the formation of a benzene ring. The enzyme that's responsible for this is known as an aromatase. Aromatase, A-R-O-M-A-T-A-S-E. Why do I tell you that? Well, I tell you that because some tumor cells are what are called estrogen sensitive, meaning that when estrogen is present, their growth will be stimulated. Their growth will be stimulated. So one of the treatments for estrogen sensitive tumors is to give something called an aromatase inhibitor. Aromatase inhibitor is inhibiting the production of female hormones and inhibiting the production of the thing that would stimulate the tumor growth. Okay? So this is a very important anti-cancer consideration right here on the right side of this screen. Okay? As you can see, there are 5 categories of steroid hormones. Okay? Besides the sex hormones, the androgens and the estrogens. And by the way, the things in parentheses are the general categories. Androgen is the category, testosterone is a specific one, estrogen is the category, estradiol is a specific one. Okay? Glucocorticoids is a category, cortisol is a specific one. Glucocorticoids are involved in inflammation, swelling, sometimes pain type responses. Okay? Progesterones are involved in maintenance of pregnancy. Maintenance of pregnancy. And the mineralocorticoids, of which aldosterone is an example, are involved in balance of ions. They help to balance sodium, potassium very well in your body, something that needs to happen. So all of these are made ultimately from cholesterol. Okay. Going from there leads me to the vitamins, and I want to talk about vitamin A second. I first want to talk about these guys, the vitamin D, E, and K. There are 4 fat-soluble vitamins, vitamins A, D, E, and K. And one of those, vitamin D, is actually made from a cholesterol-like compound. So cholesterol is a precursor of vitamin D. Okay? This molecule called cholecalciferol is, by the way, vitamin D is a very confusing vitamin. People talk about the active versus the inactive forms of vitamin D. And you can't take the active form of vitamin D. You take a precursor of that called vitamin D3. Your body pretty carefully regulates how much active vitamin D it maintains, because vitamin D is very important for importing calcium and potassium, which is important for bone balance. And importing too much of those causes big problems. Kidney stones, okay? That can be a problem, all right? You can have calciferation of various tissues that can be problematic. So the body controls pretty carefully how much active vitamin D is there, so you don't import too much stuff. Well, one of the ways that you make vitamin D from cholesterol involves light, sunlight. Ultraviolet radiation actually breaks a bond. That's why vitamin D doesn't look like cholesterol, because it breaks one of those ring bonds in cholesterol and opens up this structure to look like this guy right here. So vitamin D, when we say people should go out and get sunshine, the reason they should get sunshine is they go out and they make vitamin D by UV catalysis of that. Now, vitamin D has been a very investigated vitamin in recent years because we begin to realize vitamin D is very important for the innate immune response. And what we find, the more people we look at, is it's a vitamin that many people are deficient in and they have no idea. You might say, oh, I break my bones easily or something. It doesn't work that way, okay? You worry about osteoporosis, for example, okay? And nobody out here is sitting here worried about osteoporosis, I'm guessing, because none of you have broken any bones. And you say, oh, that's an old person's disease. And it is. But do you know when it starts? Right now. Right now. Osteoporosis doesn't happen because you're deficient in vitamin D for a week or a month or a year. Osteoporosis happens because people have problems with absorbing calcium and phosphate starting in their younger years and accumulating that over the years. If there's a time you want to be monitoring your vitamin D, now is the time. Okay? Now, I'm not saying everybody should go out and start taking vitamin D. No. Okay? But I'm saying that when you go to the doctor and you get your blood checked, and doctors are increasingly do this, they should be watching your vitamin D levels, especially young people, especially young women. You're more likely to suffer from osteoporosis when you get older if you're female than if you're male, although males certainly have the problem as well. Okay. So you see 2 forms of vitamin D up here, the active form, and I can say I use that in sort of quotation marks because the active form is not something that we can actually take. Okay. Alpha tocopherol, vitamin E. Vitamin E is very important for what we think are protection purposes. Protection, okay? Reactive oxygen species that I mentioned last time can damage things in membranes, and they can damage a lot of things. And so our body has a variety of mechanisms for protecting against reactive oxygen species. One of the things I talked about last time was the cell can commit suicide if damage is sufficient. Vitamin E helps to protect membranes from damage by reactive oxygen species. So it's a preventative for that. Now, vitamin E is the one we know the least about. A vitamin E deficiency is almost unknown. I won't say it's completely unknown, but it's very hard to be deficient in vitamin E. Okay? Vitamin K, on the other hand, is important for another purpose. And vitamin K is important for the process of blood clotting. Okay? Important for the process of blood clotting. The recycling of vitamin K, in the process, well, the way that vitamin K stimulates blood clotting is that vitamin K favors the alteration of prothrombin. It's one of the precursors, it's the precursor to thrombin, which is necessary for the clotting process. And that activation of prothrombin requires vitamin K. Activation of prothrombin requires vitamin K. Vitamin K, when it alters prothrombin, becomes something different from vitamin K. Okay? And so that has to be recycled back to vitamin K if the body is going to continue the clotting process, or at least efficiently continue the clotting process. You begin to see, well, maybe there's another thing we can have with blood thinners here, and there is. Compounds that inhibit the recycling of vitamin K also act as blood thinners. And one of these you know as rat poison. Warfarin is a blood thinner. It inhibits the recycling of vitamin K. It's a rat poison because giving it to rats, mice, anything in high doses causes them to hemorrhage and they die. Now people say, oh my God, I'm going to find all these bloody rats or bloody mice around my house or whatever. No, hemorrhaging leads to internal bleeding and they die from that. Now some of you may have heard of warfarin because it's not just used as a rat poison, it's also used to treat people who have clotting problems. It's not uncommon, okay, for people, okay, to get treated with warfarin. It's called a variety of names, but it's the same compound. And it must be very carefully dosed to make sure you don't give too much. You want to inhibit a little bit, but you don't want to completely inhibit because what's going to happen if you stop clotting? You're going to hemorrhage just like the rat, right? So people have to get monitored for that fairly carefully. They usually go in about once a month to get their blood checked to see if that happens. Was that your question, or did you have a different? Heparin is actually in a different category. So let me save that. If you want to come by, and I'll talk to you about that. Okay? Okay. Other questions? Yeah. Prevents the recycling of vitamin K. So once vitamin K gets used once, if you don't recycle it, then once is it, right? So you have less vitamin K active if you have some of this compound present, so you have less recycling, and therefore you have less clotting. That's why it's called a blood thinner. Okay? Good question. Okay. Oh yeah, question.
So it prevents recycling. Does it prevent recycling directly, or does it inhibit the enzyme associated with the recycling?
So does it inhibit the recycling directly, or does it inhibit the enzyme associated with recycling? It's the latter. It inhibits the enzyme that's involved in the recycling. It's actually an enzyme called an epoxygenase that makes an epoxy bond that's necessary for that to happen, but it's inhibiting the enzyme. Yeah. Okay, so there's your DEK vitamins. I said I'd talk about vitamin A next. Vitamin A, of course, everybody knows is involved in vision. And as you look at this picture, you can see there's actually 3 different forms of vitamin A that we find in our body. Okay? They're called retinol, ending in an OL. That's the alcohol form. And that's the form that is largely stored. It's stored mostly in your liver. And it's stored oftentimes as an ester. Okay? You notice it says all trans. All those bonds out here are in the trans configuration. And that's the most common form of vitamin A that we have. The aldehyde form, you see we have an aldehyde here instead of an alcohol on the end. And you notice a cis bond. You see a trans and a cis. Okay? This aldehyde form is the form that's found in vision. Because of that conversion of the trans bond at position 11 to the cis bond at position 11, that simple thing is the reason you see everything that you see. Everything happens because of that guy right there. How does that happen? Well, it turns out that that bond at position 11 is sensitive to light. Light catalyzes that interconversion. Okay? Now, I like to draw this analogy to a straight leg versus a bent leg. Okay? And we can imagine that a straight leg versus a bent leg poses a different structural consideration. And this guy turns out to be in the middle of a protein. And that changes the structure of the protein. And by changing the structure of the protein, we change the response of a nerve cell in our retina. So when this guy goes from this to this or back to this, okay, that is happening because light is present. And because light is present, that nerve cell is getting stimulated. It tells the brain, hey, I saw some light. And the brain takes all of those signals and paints the amazing thing that you see as vision. It's an incredible thing. Not only do you see that light happened, you see color. Unless you're colorblind, you see color. And that's a more complicated process that I won't go into here, but it involves the same thing, just different proteins that are involved in that process. Now I want you to remember the straight leg versus the bent leg, because I'm going to show you later in the class how to make a photosynthetic fish. And it involves vitamin A. Yes?
So I've read a lot about vitamin A causes the cell to mature.
Cause the cell to be mature? Yes,
So I was wondering, for cancer cells,
Yes.
cancer cells are more active than they are immature. So I was wondering, why is it not treatment?
Okay.
More vitamin A to the cell mature?
Okay, so very good question. So his question is, vitamin A is involved in the process of cellular differentiation, which is what you're calling mature. And why is it that we don't take vitamin A to treat cancer? There are actually a couple cancers where that's done, but you don't want to do it on a widespread basis. And I won't go into the reasons for that. But suffice it to say that the form of vitamin A that you're asking about is not the alcohol, it's not the aldehyde, but it's a form known as retinoic acid, which is the acid form of this. Okay? And there are some cancer treatments that involve just what you're talking about. But it's a more complicated process than this would just cure cancer. It doesn't happen that way. But some cancers are actually treated using that. Okay? Good question. Very good question. Okay. Where do we get vitamin A? Well, again, these vitamins we don't synthesize. The only vitamin that we synthesize is vitamin D. We get these from our diet. And in our diet, we get beta-carotene, which looks like this. We break it in half, we get 2 vitamin A's. Turns out we only get 1, but it looks like we get 2. Okay? And we get beta-carotene from vegetables like carrots. Green leafy vegetables have a lot of beta-carotene in them. Okay? And so we have abundant sources of these. Vitamin A is one you want to be very careful. You do not want to take too much vitamin A. Okay? No, you won't get too much from eating vegetables. That's the good news. You should not eat, you don't want to eat vegetables, right? That's what it is. Mom, I don't want to eat these peas because I'm going to get too much vitamin A, right? It doesn't work that way. It turns out you cannot overdose on vitamin A with beta-carotene. Oh, damn. The meat eaters are going, shit, right? All right. The way you get too much vitamin A, there's 2 ways I know of. One is to start taking vitamin A tablets, and it's even hard to do that because they've now made it so low. But vitamin A is toxic to the liver in high doses. The other way you can get too much vitamin A is by eating polar bear liver. There's so much vitamin A in polar bear liver that you can overdose on it. So if any of you are planning to go north for the winter, stay away from polar bear liver if they serve it anywhere, because you can actually get too much vitamin A. Okay. Did I just shoot your balloon down there with that? Okay. All right. Let's turn our attention to membranes. Membranes. Everybody knows what a membrane is. Membrane is the barrier between the cell and the rest of the universe. Everything that we know happens inside of the cell happens because the membrane provides an enclosure that separates that cell. Many of the molecules I've talked about so far, not all, but many of the molecules are found associated with membranes. The membrane consists of what we call a lipid bilayer. And I'll show you that up close. Actually, you can see it on the top thing here. The lipid bilayer contains 2 layers, a top layer that you see here and a bottom layer that you can see down below. And these are oriented so that the nonpolar portions of these molecules, remember these are all amphiphilic, partly polar, partly nonpolar, like the glycerophospholipids are a really good example. Okay? These amphiphilic compounds arrange themselves so their nonpolar portions are pointed inwards and the polar portions are pointed outwards. So for example here, top might be outside of the cell and bottom might be inside of the cell. And on either side of the cell, that's where you're going to find water. And that makes sense because the hydrophobic things are going to avoid water by associating with each other, and the hydrophilic things are going to associate with water. Okay. Here's another view of the same thing, and you can see some of the structures that lipid bilayers can assume. A liposome, a micelle. Now, I don't like this micelle figure because it shows two tails coming off of there. It's hard to make a micelle with glycerophospholipids because of those two tails. They're too bulky to form micelles very well. What forms micelles well? Soaps and detergents. They've only got one tail. That's why you wash your hands to get rid of grease, because they're forming micelles that grab the grease inside of there and pull it away. They don't have two tails, they have one. Okay? The two tails tend to favor a lipid bilayer, more like what we see with the liposome here. By the way, I should talk about liposomes. Okay? Liposomes are a very cool way of delivering drugs to cells. The lipid bilayer is a great barrier. It's really great barrier. Things don't move across the lipid bilayer. Most things don't move across the lipid bilayer very well at all. There's only a handful of small molecules that make it across the lipid bilayer very well. You should know what these are. Water. Water makes it across the lipid bilayer fairly readily. Carbon dioxide makes it across the lipid bilayer fairly readily. Oxygen makes it across the lipid bilayer very readily. And here's the bad one, carbon monoxide also makes it across the lipid bilayer very readily. Carbon dioxide, oxygen, and carbon monoxide have very similar structures. Okay? Most molecules don't make it across very readily. Glucose, horrendous at making it across. Glucose doesn't make it across very well at all. A proton, a single positive charge, does not make it across very well. And if that turns out to be really important for us when we go to generate energy. So the bilayer is an important barrier, and I said the liposome was important for getting drugs in. How does that happen? Well, imagine I got this drug that is really good at stopping DNA polymerase. And I want to kill this cell that's rapidly dividing. It's a tumor cell. And I've got this DNA polymerase inhibitor. Well, how do I get it into the cell? Right? The drug isn't going to cross the lipid bilayer very readily. How do I get it in there? Well, if I just treat the cell, it's just going to sit outside the cell and do very little. The cell is going to continue to grow, et cetera, et cetera. But what if I take a bunch of glycerophospholipids and I put them in an aqueous environment like a glass of water, okay? And I put my drug in there and I shake them up. What's going to happen? Well, it turns out the glycerophospholipids will spontaneously form liposome-like structures because those nonpolar portions are going to avoid water, and they'll make a structure like an artificial cell. If I take that artificial cell and I mix it with cells, do you know what will happen? They will fuse. And when they fuse, guess what gets squeezed into the cell? The drug. Now, it's not commonly done, but it's a way of introducing compounds into a cell by using the properties of a lipid bilayer. Okay? An artificial lipid bilayer, a liposome. Okay. Let's talk about, remind us about phospholipids. Phospholipids included things like the sphingomyelin, like the glycerophospholipids. They had a polar head and they had a nonpolar tail. The other sphingolipids, like gangliosides, cerebrosides, will have a polar head as well and a nonpolar tail. And so all these compounds readily, spontaneously form lipid bilayers when placed in an aqueous environment. Okay? You see, I've drawn this as a bent tail. Why do you suppose I did that as a bent tail? Remember I said that most commonly fatty acid number 1 was saturated. That's the straight guy. And fatty acid number 2 was most commonly unsaturated. That's the bent guy. The more bending you have, the lower the freezing temperature. The more you see of this stuff, the lower the freezing temperature, meaning that your membranes are more fluid down to a lower temperature if you have more unsaturation in the fatty acids of the glycero and sphingolipids. Okay. Now, phospholipids are distributed unevenly across the membrane. And I'm not going to ask you to regenerate this, so just showing you this to demonstrate to you or illustrate to you that if we look at the outer layer, that is the portion outside of the cell or outside of the organelle, and we compare the composition of these various lipids here, we see a distinct bias in how they're organized. Some of these, like cardiolipin, which I don't think is even on here, cardiolipin has a function that we saw associated with apoptosis, cellular suicide, okay? And each of these will also have their own functions associated with them. In some cases, they're not known. But the cell doesn't randomly put these things together. The cell has a bias by which things are put together. Nope, I jumped too soon. If we look at individual membranes of the cell, the Golgi apparatus, the intermitochondrial membrane, look at the intermitochondrial membrane. You'll remember that's where I said cardiolipin was abundant. Cardiolipin's in blue. You can see it's most abundant in the intermitochondrial membrane because of that role I told you last time in signaling that damage from reactive oxygen species has happened and the release of cytochrome C to stimulate cellular suicide. Okay? We don't see cardiolipin present in, for example, the endoplasmic reticulum. We don't see it in the nucleus. And we see it in pretty small quantities in most other places. So that function that it has in the intermitochondrial membrane is why we see so much of it in the intermitochondrial membrane. So again, cells are very careful in how they organize their membranes with these compounds. Look at cholesterol right here. Okay? Cholesterol in purple. Lysosomes, plasma membrane, look at that, okay? A lot of cholesterol in those places. All right, cellular structures. Now if we take this membrane and we start examining the membrane, we discover that there's more to the membrane than the lipid bilayer. The lipid bilayer has things embedded in it that are important for cellular function, okay? Commonly we see things, here's cholesterol. You can see cholesterol embedded in the lipid bilayer. And you see cholesterol only goes into one layer at a time. It doesn't span it. We see one over here, right? Because it's only big enough to go through one side of the lipid bilayer. We also see within here, we see things like proteins. Okay? Proteins are very common in the lipid bilayer. And why is that the case? Because the lipid bilayer is so good at keeping everything out. If it's so good at keeping everything out, how does the cell eat? Where does the cell get its energy from? I told you glucose doesn't make it across the membrane very well by itself. That means you have to have specific proteins that let in and let out specific molecules. And these proteins are embedded in the lipid bilayer. The ones that are involved in transport, like transporting glucose or transporting ions or something, are integral, meaning they go through both layers of the lipid bilayer. That makes sense, because if it's going to grab glucose out here and deliver it into here, it darn sure better go through both sides, because otherwise it would be just dumping it into the lipid bilayer itself. That would do no good. Okay? Okay, another thing that we see here, and I did this the wrong way, the outside of the cell is actually top and the inside is bottom. And the reason I know that is by the orientation of these oligosaccharides. Oligosaccharides are what I like to call license plates for the cell. They give cellular identity, they give organismal identity. The pattern of oligosaccharides on the surface of your blood cells is responsible for making the A, B, O blood types. And that's determined by these guys. Okay? Organ rejection happens because of an organ having the wrong type that's transplanted into a person. Okay? And all of the oligosaccharides are oriented outwards. They're not oriented inwards because of that license plate function. Okay? If you had your license plate you kept in your glove compartment, that wouldn't serve a very good purpose, at least as far as the police is concerned. It might be good for you if you're robbing a bank, but as far as the police is concerned, that wouldn't be good. That license plate needs to be seen. Another thing we see about this is that the internal portion of the lipid bilayer is very nonpolar. When we have proteins that go through those, the portions that touch that nonpolar part themselves have to be nonpolar. So this protein that winds through here, this portion of the protein would be nonpolar, and the portions on either side that stick out would be polar because they're associating with water. And that turns out to be an important consideration in identifying proteins that are involved in various things. Okay? Questions about that? All right. Well, I'm running out of time, so I want to mention one last thing before I go to our song for today, and that is the movement of materials across the lipid bilayer. You saw that there was a bias for certain molecules on the inner versus the outer portion of the lipid bilayer, and you saw a bias for certain organelles that had some of one kind versus another kind on the inner versus the outer. And the question then is, how do these molecules get to where they're going? Okay? Well, they don't happen randomly. There are two types of motion that are associated with lipid bilayers. One's called lateral movements, and that involves movements within a single layer. You can think of it as left and right. So if I put a molecule here on the very top and it moved left and right, that would be lateral. So I could do an experiment, for example, where I took a group of molecules and I labeled them in some way, so that you see here at the top they're all black. I might use a laser to make them all black. And then over time, I measure how long it is until they get completely mixed into the cell. And when we do this, we discovered, okay, here's 100 seconds, here's 200 seconds, that's 3 minutes. That in 3 minutes, these molecules completely disperse across the cell. What does that tell us? It tells us that the lipid bilayer is very, very fluid. Okay? It's something we call the fluid mosaic model, meaning that a membrane is not something that's fixed, but that there's a lot of lateral diffusion. Now there's another type of diffusion that we can think about, and that is not moving within a single layer, but flipping from one side to the other side. That's called transverse diffusion. Okay? Transverse diffusion happens as a result of action of an enzyme. Transverse diffusion does not happen by itself, hardly at all. Enzymes that have really cool names catalyze the movement of molecules from one side to the other. The first of these to be discovered were called flipases. Okay? They moved molecules from the outside to the inside. Then they discovered a category that moved from the inside to the outside, and they called them flopases, which I kind of loved. And then they found one that does both, and they called it, what else? A scramblase. Okay? But all of these enzymes are involved in transverse movements from one side of the lipid bilayer to the other. Okay, that's a lot for today. Let's turn to our song. And today, remember I said that you get extra credit if you sing loudly? Today I'll be able to measure because you're going to do most of the singing. I'll help you, okay? But I don't have somebody helping us with this one today. It's called The Fatty Acids in Our Cells. It's a very easy song to sing. And for those of you that are leaving, there's no need to leave. You can just hang around because class isn't done yet. Okay, pop quiz. They think I'm kidding. All right. Fatty acids in our cells. Let's go. So, from the fatty acids in our cells to the lipids in our brains, we are made of biochemicals built in metabolic chains. Using glycolytic ATP and electron energy, we can synthesize most everything with the help of delta G. I can't hear you. A cell will tend to pump out sodium, but potassium it imports. It accomplishes this magic with ATPase antiports. Our bilayer lipid membranes protect the cell's insides. partly made of sphingolipids we know as gangliosides. When it comes to regulation, the little cell has got it made. It phosphorylates a lot of things with its own kinase cascade. Stimulated at a hormone site, metabolic yang and yin. That's turned on by epinephrine and turned off by insulin. All right, guys. Woo!